just a moment to tell a little bit about my situation, because I was also last week in the World Congress and I was one of the unlucky ones. So I got corona from there. So I might need to stop time to time to sneeze or cough my lungs out, but hopefully it will go well. So let's start. So first, something about the Viking genetics, where I have been working for the past 4 years. So we are a cooperation in the Nordic countries. So this includes Denmark, Sweden, and Finland. And we are a cooperative owned by the 17,000 farmers in the Nordic countries. So we have also 3 daughter companies in Australia, Germany, and United Kingdom. And we have export on 50 plus countries, and I think Italy is also one of the countries where we do some of the export. We have our main office in Denmark. So currently I'm also living in Denmark. I just moved there in April because Arhus University is also very close where we have the headquarters in Denmark. But at the moment, because I'm in a summer holiday, I'm approximately here, middle of Finland. So the Viking Holstein is the breed of what we are breeding, and we have a common Nordic breeding program. So we have the same breeding program in all of our 3 countries. And in total number of Holstein cows at the moment, we have approximately 500,000, almost 600,000. And mostly we have the cows in Denmark, but quite a lot also in Sweden and Finland. Sweden and Finland also have quite large populations of Nordic Red, what has been mentioned earlier today in some of the presentations. So it's a combination breed of the Finnish Ayrshire and Swedish Red and Danish Red. And currently we genotype approximately 70,000 animals in Holstein per year. But this is also increasing very fast. Like last year we had 70,000, but the year before that it was around 40,000. So it is going up very, very fast. And average milk yield on the Holstein is 11,000. And here is some of the information about the proteins and the fat contents on the milk, what they averagely have in the Nordic countries. So let's go to this topic of inbreeding. Well, we could say that we have 2 types of inbreeding. We have the long-term inbreeding and also the short-term inbreeding. This has been mentioned earlier today, that this short-term inbreeding, what happens in the past 5 generations, that these tend to have the more negative effects on different traits. And the long-term inbreeding seems to have more neutral or even positive effect, because that is the inbreeding what has been under selection. So we have been actively selecting against of these signs of the inbreeding depression. So in a simple way, inbreeding is mating between individuals who share common ancestors, but we should try to minimize the short-term inbreeding more, maybe in the dairy cattle. So what causes inbreeding? Well, many times it's the environmental changes, changes on the breeding goals. Popular size is one of them. And well, I think environmental changes are a little bit more about on these levels of war times or pandemics or anything like that. This seems to be happening now all around the world, what is affecting these things. And in the recent years, many of the populations have gone through the popular size. So we have been using minimum number of sires to many of the females. So they have caused this genetic bottleneck, which might have caused lead us into the inbreeding depression. Sorry about that. So in short terms, the inbreeding depression causes reduction on milk production. It increases the calving interval and increases somatic cells and decreases your fertility. This was done. The study was done in the Netherlands, and I think there has been after this there has been a few other studies which have been confirming these findings in Holstein and also with some other main dairy breeds. So in Viking genetics, we have taken actions to control inbreeding for a quite long time, and the aim has been to maximize gain while restriction increase of inbreeding. And we have been focusing on the short-term inbreeding in our farm level matings, because we have quite a lot of control of breeding in the farm level in Nordic countries, because our owner companies control about 90% of the market share. So we have the breeding plans are done by the breeding companies in each of the countries, Faba, Vexa, and Viking Denmark. And there we have a limitation of inbreeding, that the inbreeding level when we calculate from the 7-8 generation, it shouldn't go over 4%, for example. So we have different softwares developed on this type of breeding planning. And then in the long term inbreeding, we are trying to control that in Viking Genetics as a breeding company. We use optimal contribution selection when we are selecting the animals of bulls for purchase or the heifers for embryo production. And we also have a very multi-trait breeding values. We have had used health traits for a very long time, and I think our health program is one of the longest one in the dairy cattle world. So how well have we managed inbreeding? There is a typo in the Nordic. So we did this small study for the World Congress, and I wrote a paper. Some of the results was actually shown in the first presentation this morning. But here you can see the calculations, what we did with the Holsteins. And these are the, we have here is the average inbreeding level, expected inbreeding level, and average co-ancestry in the Nordic Holsteins. These trends are based on the most recent generations of females and their trace pedigree. So it doesn't include, for example, animals which have the lines which have died, for example, in this time period. So this tells us that in this time we have also quite a large increase on the generation interval, because before genomic selection was implemented, our generational interval was almost 5. But nowadays it is this is on average between the females and the males, by the way. And nowadays it's about 3, and I think it has been decreasing from that. And we can see that our inbreeding trends has been exactly the same before and after the genomic selection has been implemented. This is a little bit different what we have seen in many other dairy productions and dairy populations in different countries. And I hope I can give you some answers on why this happens later on this talk. But because we have a decrease on generation interval and also our inbreeding trend has been the same, this has by calculating, this has caused us that our effective population size has increased from 70 to over 100. But it's quite a nice, and we hope that this trends goes on, because here you can see this trend was calculated until 2018. This is actually when I started to work in Viking Genetics, so this was actually one of the first projects what I did. So these are now, hopefully we will be updating these very, very soon. Because also in this time after 2018, I started to work to implement optimal contribution selection on our monthly selection processes, and hopefully that has changed some of the trends. But here you can see also that in the same time when we have managed to keep the inbreeding trend stable, our genomic trend in Nordic Holstein has gone up. Well, the female trend has been quite stable, but the males, we can see that there is quite large increases over the time. And well, it seems to be speeding up on the end of the year. There might be some connections, because here we can see that there is some increase on co-ancestry that maybe in these times we have been starting to use more related animals, but hopefully we will find an answer to that after I get new updated calculations done. Now I need to take some water. So before we started to use genomic selection, we did lots of simulation studies on how many animals and how well we should select these animals. And before we started the genomic selection, we used much more bulls than we use today. And the simulation studies gave us an optimal number of bulls what we should select per year to still keep a good progress on genetic gain, but still keep the increase of inbreeding in a good level, so it doesn't go up too fast. And we also, by these simulation studies, we increased the number of females in the reference populations, and then we also decrease the number of bulls. And here you can see some of the progress on what we had with the bull numbers over the years. So as you can see, we had, before genomic selection was implemented, we used over 300 bulls, and after the genomic selection, we are around very much low. And currently we are in this 2018, I think we were still buying about 100 Holstein bulls per year. So about 80 of them were used for breeding. And there is the part of the foreign bulls, because we also buy bulls from outside of Nordic countries, for example, from Germany and France. So there is always, and then we also import some semen from, for example, US or Canada. And here is also the line of these Saija sons. And well, number of Saija sons has been quite low in the beginning compared to the size number of bulls what we have been using. So this means that we had also very many bulls from the same sires. So these, after we have done the changes, this number is pretty much the same, because we have an aim on the breeding program that all the bulls what we use, what we select for semen production, that they will produce bull offspring what will be selected to continue his work in the semen production. So that's why these numbers should be pretty much equal. Here is some information about what is the Viking Holstein breeding program at the moment. So we are selecting, we genotype about almost 3,000 bulls per year. And we also genotype quite a lot of females. And we buy 70 bulls, and approximately 60 bulls of these end up in production. There is always some fail rate. And we also focus heavily on the embryo program, because currently we have seen this trend that more and more Holstein bulls or our breeding bulls are born from our embryo programs, because more and more farms are using sex semen to their best females and beef semen to the not so good females. So there is this kind of change on the dairy production in the Nordic countries. So that's why we put lots of focus on that we do the correct matings in the embryo production. And that makes it a little bit easy, because in embryo production we can also use all of our own bulls. So we are not so depending on what the farmers are doing. And well, a little bit about the current NTM, or Nordic Total Merit Index. So we have 15 main traits and more than 90 sub-traits. This includes the newest one is saved feed, and there is lots of health traits. Here is about percentage which kind of traits these 90 traits are. And we have been including the health traits for quite a long time, and I think this has been also one of the factors why our selection of bulls have been a little bit more diverse compared to many other countries. So summary of this study, this small study, so inbreeding and co-ancestor trends has been the same before and after genomic selection was implemented. Generation interval has decreased, and the effective population size has also increased to this. And we have also decreased the number of sires what we have been using, but number of sires of sons have increased, or it has been very much equal to the sires what we have been using. And based on the statistics in NAV, Nordic Genetic Evaluation Center, genetic gain has increased after the genomic selection. So this was just a starting point for my work in Viking genetics. So how we manage inbreeding now, and this will be the big part what I have been working in the past 4 years, and which have led me to start my PhD. So this might look lots of arrows, but this is how our optimal contribution selection scheme works. So EVA, what you can see on these green boxes, that is the program what we use to do optimal contribution selection. And these are all the selection steps where we try to use it in the most optimal way. It's not an easy system, and it takes quite a lot of coding and quite a lot of testing to get it done. So I started to develop this in 2018, but the pre-work has been doing, they have been doing it for like a decade before me. So I just came in the end and did the fine tuning and how to implement into the practical use. So a little bit about the different steps. So in the beginning of every month, we get the breeding values. So then we start to select which bull calves we should buy. So these are the bull calves which get their genomic breeding values, and then we use them as a candidate to see which will be the most optimal young bulls to our young females. And after that, we go to the selecting which females we should select for our embryo program and which females we should, for example, put on embryo on these field flush contracts. These females, which are field flush contracts, they are normally something what we might find interesting based on their pedigree, or they have some traits that we are interesting, but their genomic level might not be the top of the top, but we still find them interesting. So we give them this contract that they will be mated to one of the most recent and best sires, so we can get embryo offspring from them. And then we start screening on these high index females. These are what we, and also which sires or sons we should keep and which should be used for these females. So we select every month with HOLSTEIN, for example, we select these dams. There is about 10,000 dams selected per month. Oh, the total number of them is 1,000, but approximately a month there is 1,000 more what we select into that group. So all genomic bulls are used as sires or sons. And so first semen goes to the ET and OPU programs and to these potential bull dams, and also into our embryo program. And here is also marked that 5% of this semen what we use on these steps is imported semen. So this means that the 5% comes from US or Germany or some other country. We have been quite much using French some time ago. And then we also do these screenings of pregnancies, and we try to find which soon-born calves we should genotype, because we all know that there is very much an urgency when the calf is born, the bull calf is born, and we want to genotype it very fast, even if it's in a farm, because the farmer doesn't want to keep that calf very long, or it will sell it to these meat rearing stations or bull rearing stations. So we try to get them genotyped very fast and get the results also. And well, we do this on pregnancies. So those bull calves which are not the calves which are not bulls, they are female, so they will be genotyped also, but they are genotyped for the heifer or donor program. So the future plans are that. Well, first of all, I started my PhD project. So this is a little bit on the balancing. The project name is Balancing Genetic Gain and Diversity in Dairy Cattle Breeding Schemes in Genomic Era. And we started this basically because of my work in the past 4 years, because we noticed that we are now doing all of this we are doing with pedigree information, and we know that we might need to look into the genomic information, especially when we nowadays see more and more results from random homozygosities and stuff like different how to do the selection better. So this is a cooperation project between Viking Genetics and Arhus University, and my supervisors is Per Berg and Modern Cargo, most likely very familiar names. Actually, Modern Cargo was supposed to give this presentation instead of me, but he decided to go to summer holiday. And well, for this project, the aim is to find an optimal way to estimate inbreeding and relationships, especially on the relationships. We know that the random homozygosities work quite well on this diagonal when we know the individuals inbreeding, but how to estimate which is the best way to use it off diagonal? Because many times when we do breeding plans, we want to know how related two animals are. And well, the main aim is also control inbreeding in an effective way in the Nordic dairy cattle populations with the genomic OCS. Sorry about that. And well, the aim should always be that we want to have genetic gain, and we don't want to be pushed out from the market because we are too much focused on inbreeding. So we also have quite much of pressure on keeping the trend going up with genetic information, and also that we don't lose too much variation on our breeding animals, because we are all the time adding new different traits into this total index. And well, sometimes it might go that we have lost some of the variation what different traits might need, and we don't want to end up in that situation. And well, new changes in the breeding program, and what I said earlier, it's the movement towards this nucleus breeding. So we have very close breeding cycle. So the breeding of the breeding bulls is happening on these embryo matings, what we are doing in the company. So there will be no bulls born in the commercial farms. We already see this. This have already happened in the Jersey in Denmark. So there is like few % of bulls born in the commercial farms. And most of them are now coming in from our breeding program in the embryo stage, and hopefully we will do that selection optimally, because Jersey is also a very small breed, and if we lose too much variation there, it might be quite crucial. So I would say thank you, and if you have any questions, feel free to ask. This might be a little bit shorter because I'm having problems on talking.
Thanks a lot for the presentation. I think we don't have questions on the online. Is there any question here in the audience? You have several questions already here.
Thank you, Saija. Thank you for your nice presentation. I have a question related with the last slides that you show us. You decide to move on a closed nucleus breeding program, yes, without using commercial farm. So can you explain better why?
This is partly because, for example, in Denmark, there came this law that you're not allowed to kill bull calves when they are born. So this was the driver why Jersey breeders moved towards using beef semen, because Jersey bull calves are quite small. They are not very profitable on the beef business. So they wouldn't get any money out from if the calf that was born, it was male. So to be a bit profitable, they changed to use sex semen on the top animals and then beef to the rest. So that's why there is no bull calves born anymore, pretty much. There is like 5% because there is also the error rate on the sex semen.
We have another question. Thijs?
Okay, I had the same idea about the nucleus, that maybe if you have a closed nucleus that you would lose genetic variation. I have another question is about the crossbreeding in the Nordic countries. There's a crossbreeding, I think, between Holstein, Danish Red and Montbéliard. Is that right?
Yes, there is the Pro-Cross program. Yes.
So, and one of the main breeds in the Nordic countries is the Jersey. The Jersey is not part of the Pro-Cross. The reason...
No, not Pro-Cross, but we have a separate cross program called Golden Cross, where the Jersey is the third breed. That is very popular, for example, in New Zealand.
Yes. Okay.
So we have two crossbreed programs. So they are a little bit on different purposes.
Okay.
Thanks. Is there any Christian? He's coming here, so.
I was sure they will be from him.
Hi, Saija. Nice seeing you again. I hope I'm not the one who gave you COVID. I didn't get it, but maybe I was.
No. I think quite a lot of us got it there, but I'm getting better. But it's still on the edge.
So, very nice presentation. My question is, I don't know exactly the structure of the industry in Denmark, but you have a very elegant system, but it's a system where you have only few players that do not have conflicting objectives. So how do you see that played out in my courtyard, where everyone's saying, well, we should do that, but no one can agree to do it? So, and if you have any thought about how that something similar could be implemented on a slightly different system. Thank you.
Yeah, I know it's the political side is the much difficult side on this than what we actually do in the calculations. Luckily, I normally work mostly with calculations, so I don't need to take care of that much of the politics. Though I am in the Viking board also, so I'm also in the both sides. But yeah, I think it's more about finding the common ground and having. I would say, meetings. We have our disagreements in the Nordic countries all the time, even though we do work in the same goal. But we have also lots of meetings where we have all the 3 countries present, because the problem is also what we see inside of the countries, because we have somewhat differences on the milk price systems, for example. So in Finland, we for example get more subsidized money for the kilograms, and in Sweden and Denmark they are more focused on the proteins and the fat, what is inside of the milk, not how much you make. So this is a little bit on the conflict, and we just need to have these discussions on where to go and find the common grounds. And I think this might work in the same way no matter where you are. You just need to agree to disagree and go forward in the one or the more simple ways how to do cooperations. And I know for us it's a little bit easier because, as I said in the early stage on the presentation, that we have about 90% of the control of the breeding, the Nordic dairy cattle breeding, and most of those the Nordic dairy cattle then go also milk recording, so we have lots of data also. But there is also some which are not agreeing, and yeah, there is some changes also happening, but hopefully we can go through it. I don't know if that answers your questions, but well, I also work with dog breeding, so they're always saying about you need to talk.
Christian is saying that you answered his question.